So many of you are interested in how ivermectin might be working in cancer, and you might or might not be aware that there is a clinical trial taking place right now in California testing ivermectin in combination with another medication for triple negative breast cancer. And they just released recently some of their latest results. This is still, it's so new that this is still fairly it's unpublished data, but this information comes from a conference abstract. So this is the first information we know of any kind in regard to any results from this particular trial. And this information tells us how the authors of the study are currently trying to figure out what is the safe method and safe dose of ivermectin for this purpose. All right, I'm gonna break this down really quick for you. My name is Dr. Mikola Rashik of Neurogenomics, and let's get started. I'm in a very cool place to do this, so I wanted to show you this, which is why I'm recording here. And what the trial is doing is they're testing ivermectin in combination with a medication that is referred to as immune checkpoint inhibitor. Now, I recently did a video explaining what that is when I was talking about another paper that recently was published showing how mRNA vaccines sensitized cancer patients to that medication, meaning when cancer patients were vaccinated, if they took this medication within 100 days, vaccination helped that medication perform better, if I can explain like that. There are some limits to that. I explain in detail in that video. I explain how the medication works. And in essence, to make it very brief and very quick, I'm going to tell you that the way it works, it's an antibody that binds a molecule called PD-1. This molecule is found on T cells, killer T cells that are supposed to go in and into the tumor area and kill cancer. And cancers can grab T cells by that molecule and stop them from being able to perform their function. So when you introduce this antibody, you prevent that interaction between cancer cells and killer T cells, and that makes killer T cells free to do their function, which is attack the cancer and destroy it. It's a fantastic medication that can really do wonders. It has saved numerous lives for people that otherwise wouldn't have made it. That's how amazing it can perform. However, it comes with a dark side as well. And the dark side part of it is that for a small percentage of those people who take this medication, it creates turbo cancers. So turbo cancers are real. They're not made up by anti-vaxxers. It's a real phenomenon, but it's got something else in medicine. It's called hyperprogressive disease. This has never existed in medicine. It was never seen before until that medication specifically targeting that PD-1 molecule has come out in use. So we know of turbo cancers of maybe in the last decade or so. Okay, so this phenomenon precedes the pandemic. Check out my videos on turbo cancers if you want to understand what might be the link between the current mRNA vaccines and turbo cancers as well, because there's a lot of these rumors online. If you want to understand the biology behind that, you could check that out. All right, so that video has all the background. So if you need to learn more, check that out. Now, one of the authors involved in the clinical trial, this person is a renowned oncologist that does great research trying to understand how the immune system might malfunction in the context of cancers. And this is how this researcher, her name is Dr. Peter Lee, and his group came to discover that ivermectin might sensitize cancers to this specific medication, this immune checkpoint inhibitors, the antibody that helps to that antibody that attacks that PD-1 molecule and helps to sensitize cancers to killer T cells so that killer T cells can destroy it. I will explain all of this in future videos because it's fairly complicated, but this group of researchers spearheaded by this guy, Dr. Peter Lee, they first discovered that ivermectin can make cold tumors hot, meaning in order for this medication, this anti-PD-1 antibody to really be effective, the tumor has to be infiltrated by killer T cells. If killer T cells are not even present, it's not gonna do anything. And ivermectin does that, and it does that by making sure more killer T cells can be present in the tumor site while helping at the same time remove what are referred to as regulatory T cells, which are like suppressive T cells that shut down the immune system function. They do that on purpose to make sure that immune system doesn't go out of whack, but sometimes cancers can take advantage of that in order to suppress the immune system from recognizing cancers. And this is well recognized as well. I've also covered that in that video where I explain this finding, how the mRNA vaccines can sensitize the cancers towards that medication, the anti-PD-1 medication. And I talk about it as well in another video that discusses how targeting parasites via ivermectin might indirectly help fight cancers by its impact on regulatory T cells, because parasites also stimulate those regulatory T cells. And I also talk about this in another video where I talk about how regulatory T cells might be explaining a lot of these paradoxical, confusing observations that we see with regard to the differences between mRNA vaccinated versus non-vaccinated people in terms of the differences in all-cause mortality as well as cancer incidence. So check that out to get the background on that. But the point here is that ivermectin enhances the killer T cells function in tumor site. Those, they can also be referred to as effector T cells, and suppresses the suppressive or regulatory T cells. So that's the magic. And they discovered that through research, and that research that they did on animals, first in cells, then on animals, has resulted in this clinical trial. And now we finally have some first information that they just published. They're still recruiting people for this trial, and now we have information on the first 9 people. So what did they do? At the moment, this is such an early stage of the trial that they're still trying to figure out what is the appropriate amount of ivermectin that should be used to accompany this anti-PD-1 antibody immune checkpoint inhibitor medication. So that's what they're trying to figure out. What is the safe amount? So they're testing 3 different amounts, and what they did is the following. They took 3 patients and they put them on the lowest amount, which was still 30 milligrams of ivermectin, and they give it to them in cycles. One cycle was 21 days. That included 1 dose of that immune checkpoint inhibitor. So you only take it once every 21 days. So that's 1 cycle. And ivermectin, you take it for 3 days in a row. Then you take a break till the first week is done. Then you do it 3 days in a row, take a second break till the second week is done, and then you take it again 3 days in a row, and then you finish that 21-day cycle. And that's how ivermectin is being used for 30 days. Then what they wanted to do is they monitor in the first cycle, in the first 21 days, is ivermectin toxic? Does it produce serious side effects? So they monitor for side effects. They grade them according to severity, and if any severe side effects are observed in the first cycle, then we're done. You need to go to a lower dose. But if no side effects are observed in that first cycle, then it means, hey, these patients that we started on 30 mg, they can continue, and then they will take 3 more patients and they put them on 45 milligrams of ivermectin. So they did that as well. That's already ongoing, and they did the same thing. They monitored them for the first 21 days, the first cycle, to see are they seeing a serious issues or not. And again, 45 milligrams was fine. So 45 milligrams allowed them to now continue that second 3 people on taking 45 mgs, and they took 3 more people and they gave them 60 milligrams. And that was the maximum dose they were going to test. However, by the time they published that abstract, it was not enough time to finish that 21-day cycle. So we don't know yet whether the final dose that they will be using will be the 45 milligrams or 60, depending whether any toxic side effects might have been observed or not in that 60 milligram within the first 21 days. So that part we're still awaiting. That abstract from that conference did not tell us that, but we know that at least 45 milligrams will be used by these clinicians in that first phase of the trial. And what they're going to do then, if no side effects are being observed throughout the study, and this was several months long already that they've been observing them, so at least probably 15 cycles have been done. And if that is looking good, then they're going to now expand it to phase 1B, which they're going to have more patients going in and doing this. So that's all we know so far, that we have these 9 people. Out of those 9, they had results for 8 of these 9 patients. Out of those 8, 6 people, the disease continued to progress, meaning cancer kept growing, but 2 of them, there was some improvement. So one of them, the disease became stable, meaning cancer stopped growing, and one individual, the cancer started to regress, I believe. And that is good enough to suggest, hey, this has some potential. We will continue with this trial. So the trial will continue. We're gonna get more data, and we'll see what kind of data gets published. I was just lucky to even see someone post this abstracts in one of the comments related to one of the ivermectin videos dedicated to cancer, and I thought I would share this because I know many of you are interested. So you can see, to do these clinical trials takes a really long time. Even just the phase 1 will take minimum 6 months, could take up to a year. Then you move to phase 2, where you continue checking for efficacy. Once we know what worked in that phase 1, they will then determine what is the definite dose that they're going to use in phase 2. And then phase 2 is going to last minimum at least a year, probably longer, before you even move on to another phase. So you can see this is a really long time. But definitely California is way ahead of everyone else. And this is thanks to this amazing research that they did in the lab, spearheaded by this Dr. Peter Lee, who's still involved, like I mentioned, in this clinical trial, and testing it on mice and showing that in mice, which is, I'll explain that in the future, it definitely worked. This combination with that immune checkpoint inhibitor and ivermectin, it definitely helped to reduce tumors and help mice survive that had these type of breast cancer tumors literally injected into them. But that's for another story. I'm gonna wrap it up here. But before I go, please like, please share, and please subscribe if you haven't already. And one more thing that I want to tell you, also please check out my Patreon account for 2 reasons. Number one, I'm now running this project where I want to, with collaboration of people who are my patrons. You don't need to be supporting patron. You can be there for as a free patron, meaning you just sign up for free. And I want to collect information from any of you in terms of what you think is the best way to lead a healthy living lifestyle, and make a top list of the most essential components of what it means to lead a healthy lifestyle so good that we don't need to see doctors or see them minimally. Of course, do your checkups, but be so healthy that we don't need to necessarily rely on doctors because we're healthy. So that's my goal. What is that? Of course, we're talking about diet, exercise, and so on. And the second reason is because those who are supporting patrons, those who do pay, they also get to vote and decide which content makes it to the YouTube. And not all of my content gets to be migrated from Patreon to YouTube. So that's the supporting patrons decide what is worthy of making it to YouTube. All right, I'm gonna see you guys next time, and bye for now.